Okay, so this is the second part that I just wanted to review with you guys before I got cut off a minute ago. The ways for questions to ask ourselves, and step number 2, ask what the main idea and details are. I want you guys to look at this. Are what is the paragraph about? So you're going to think about what the paragraph is about, and you can write it down. The paragraph is about blank. We're actually not going to be writing for this strategy. You guys are going to be using a recorder, so you don't have to write anything. You're just going to be speaking your thoughts. And then the next thing is, what does it tell me about, which might give you some of the details. So I want you guys to remember that. And then places to look for the main idea are in the first couple sentences. You want to look, even the first sentence a lot of times will give you what the main idea is for the paragraph. And then you can also look at words that might repeat or be synonyms of words in the first sentence, and that will help you get the main idea. So you guys are going to have, when you're doing this, you're going to have your reading. I photocopied a section from your science textbook, and you're going to have a microphone recorder, which will be my phone for right now. And I'm going to demonstrate how it should look as you go through. So we need to remember the steps: wrap, read, paragraph. We're going to do that first. Then you're going to ask yourself, what is the main idea and details in the paragraph? And then last, put it into our own words on the tape recorder. So I'm going to start off with this first paragraph, and I'm going to read it aloud to you, and then we'll go through the steps. So here we go. Much of the cell is devoted to making proteins. Proteins are made of 20 types of amino acids that have unique characteristics of size, polarity, and acidity. They can form very long or very short protein chains that fold into different shapes, and multiple protein chains can interact with each other. This is almost limitless. The variety of shapes and interactions This almost limitless variety of shapes and interactions makes proteins very powerful. Proteins carry out many critical functions, so they need to be made correctly. So this is paragraph 1. So I did step 1, which was read the paragraph. So now we're on step 2, ask what the main ideas and the details are in the paragraph. So like I said, we can look for to the first sentence, which says, much of the cell is devoted to making proteins. So I'm going to say that that's the main idea for this paragraph. And then it goes on to talk about the different kinds of proteins and what they do. So I'm going to pick up my recorder and say, in this paragraph, this paragraph is about making proteins. Proteins are made up of different shapes, and there are many different there's many different shapes of proteins, and the proteins need to be made in the correct shape in order for them to carry out important functions. And then I'm done with paragraph 1. And we go on to paragraph 2. The nucleus is the storehouse for most genetic information, or DNA, in your cells. DNA contains genes that are instructions for making proteins. There are 2 major demands on the nucleus. 1, DNA must be carefully protected, and 2, DNA must be available for use at proper times. Molecules that would damage DNA need to be kept out of the nucleus, but many proteins are involved in turning genes on and off, and they need to access the DNA at certain times. The special structure of the nucleus helps it meet both demands. So I'm going to say, what is the main idea? Ask myself, what is the main idea of this paragraph? And again, I'm going to look at the first sentence. The nucleus is the storehouse for most of the genetic information, or DNA, in your cells. So I will pick up my recorder and say, this paragraph is about the nucleus. The nucleus is the place that DNA is stored. DNA is responsible for making proteins, and the nucleus both protects DNA and allows proteins to access DNA. Okay, and then the next paragraph. The nucleus is composed of the cell's DNA enclosed in a double membrane called the nuclear envelope. Each membrane in the nuclear envelope is similar to the membrane surrounding the entire cell. As figure 3.7 shows, the nuclear envelope is pierced with holes called pores that allow large molecules to pass through between the nucleus and the cytoplasm. So I'm going to look at the first paragraph, or the first sentence of this paragraph, to find what the main idea is, and I'm going to say the nucleus is composed of the cell's DNA enclosed in a double membrane called the nuclear envelope. So from that first paragraph and seeing that nuclear envelope is repeated a number of times, I'm going to think that the main idea of this paragraph is the nuclear envelope. So this paragraph is about the nuclear envelope. DNA is stored in a nuclear envelope, which is made up of a double membrane. The nuclear envelope has holes for molecules to pass through. Yeah. So as you can see, I'm getting not all of the details. I'm not repeating everything that the paragraph says word for word. I'm putting it into my own words, but I'm getting some of the important information that backs up what the main idea of the paragraph is. All right, so number 4. The nucleus also contains the nucleolus. The nucleolus is a dense region where tiny organelles essential for making proteins are assembled. These organelles, called ribosomes, are a combination of proteins and RNA molecules. They are discussed on the next page, and a more complete description of their structure and function is given in Chapter 8. So, the first sentence, again, says the nucleus also contains the nucleolus. So I would say the main idea of this paragraph is the nucleolus, or that the nucleus contains the nucleolus. So this paragraph is about the nucleolus. The nucleolus is part of the nucleus, and the nucleus is, a place where the nucleolus is a place that organelles make proteins. Organelles are also called ribosomes, and protein and RNA molecules are what make up the ribosomes. Next paragraph. Then again, this paragraph, I'll show you guys a picture, has a heading, so that can also kind of help us get an idea of what the main idea of the paragraphs are going to be about. But we're going to still use the same strategy. A large part of the cytoplasm of most eukaryotic cells is filled with the endoplasmic reticulum, shown here in figure 3.8. The endoplasmic reticulum, or the ER, is an interconnected network of thin folded membranes. The composition is very similar to that of the cell membranes of the nuclear membranes. That of the cell membranes and nuclear membranes, The ER membranes form a maze of enclosed spaces. The interior of this maze is called the lumen. Numerous processes include the production of proteins and lipids occur both on the surface of the endoplasmic reticulum and inside the lumen. The endoplasmic reticulum must be large enough to accommodate all these processes. How does it fit inside a cell? So, we're going to look at the first couple sentences, and I'm going to read them again. The large part of the cytoplasm of most eukaryotic cells is filled with endoplasmic reticulum. The endoplasmic reticulum, which, by the way, is repeated a number of times in this paragraph, or the ER, is interconnected network of thin folded membranes. So, this paragraph is about endoplasmic reticulum. Endoplasmic reticulum is what fills cytoplasm. It's made up of folded membranes. The membranes are similar to cells and the nucleus, and they contain the ER membranes contain lumens, which are a place where proteins are made. In the next paragraph, the ER membrane has many creases and folds. If you have ever gone camping, you probably slept in a sleeping bag that covered you from head to foot. The next morning, you stuffed it back into a tiny little sack. How does this entire sleeping bag fit inside such a small sack? The surface area of the sleeping bag does not change, but the folds allow it to take up less space. Likewise, the ER's many folds enable it to fit within the cell. This paragraph is about the shape of the endoplasmic reticulum membrane. It has many creases and folds that allow it to change shape and to fit into different spaces. The surface area of the endoplasmic reticulum does not change, but the shape does. Next paragraph. In some regions, the endoplasmic reticulum is studded with ribosomes, tiny organelles that link amino acids together to form proteins. Ribosomes are both the site of protein synthesis and the active participants in the process. Ribosomes are themselves made of proteins and RNA. After assembly in the nucleolus, ribosomes pass through the nuclear pores in the cytoplasm where most protein synthesis occurs. This paragraph is about ribosomes and where they occur. Ribosomes are on the endoplasmic reticulum. They are they link amino acids together, and amino acids are what help form proteins. Ribosomes are also a place where proteins are synthesized. Okay, next paragraph. The surfaces of endoplasmic reticulum that are covered with ribosomes are called rough ER because they look bumpy when viewed with an electron microscope. As a protein is being made on these ribosomes, it enters the lumen. Inside the lumen, the protein may be modified by having sugary chains added to it, which can help the protein fold or give stability. So, I'm going to look again at the first sentence of this paragraph. As we go over the steps, we're going to ask ourselves what the main idea is about. The surface of the ER, endoplasmic reticulum, that are covered with ribosomes are called rough ER because they look bumpy when viewed with an electronic microscope. This paragraph, I would say, is about the rough endoplasmic reticulum, which, let me restate that, the surfaces. So here we go. I would pick it up and say this paragraph is about ribosomes and how they form rough endoplasmic reticulum. The rough endoplasmic reticulum is called this because it looks bumpy, Proteins made on the ribosomes enter the lumens, and the lumens can add sugar change to proteins for stability, so they can help change the shape of it. So again, we're going back to talking about proteins being an important part of cells and how they need to be formed correctly to work the right way. So kind of tie it back to the beginning to help remember what we're learning about. So we'll go on to the next paragraph. Not all ribosomes are bound to the endoplasmic reticulum. Some are suspended in the cytoplasm. The general proteins made on the endoplasmic reticulum are either incorporated into the cell membrane or secreted. In contrast, proteins made on suspended ribosomes are typically used in chemical reactions occurring within the cytoplasm. So, for this paragraph, I'm going to say that this paragraph is about where ribosomes are bound to and located. So they don't always exist in the endoplasmic reticulum. They can also exist in the cytoplasm, and they can be made on endoplasmic reticulum or suspended. This next paragraph is short, so we might not have as many details about it, but we'll go through it. Surfaces of the endoplasmic reticulum that do not contain ribosomes are called smooth ER. Smooth ER makes lipids and performs a variety of other specialized functions, such as breaking down drugs and alcohol. So we're going to look again at the first sentence. Surfaces of the endoplasmic reticulum that do not contain ribosomes are called smooth ER. This paragraph is about smooth endoplasmic reticulum. Smooth endoplasmic reticulum does not contain ribosomes, and it makes lipids, which are used to break down drugs and alcohol. All right, we have a couple paragraphs left to go, and then you guys will have some time to practice on your own, or with me helping you, but do it by yourself. So the next paragraph again has a heading, which can kind of help us get an idea of what the topic and main idea of the sentence is going to, or the paragraph will be about. The Golgi apparatus is the title. From the endoplasmic reticulum, proteins generally move to the Golgi apparatus, shown in figure 3.9. The Golgi apparatus consists of closely layered stacks of the membrane-enclosed spaces that process, sort, and deliver proteins. Its membranes contain enzymes that make additional changes to proteins. The Golgi apparatus also packages proteins. Some of the packaged proteins are stored within the Golgi apparatus for later use. Some are transported to other organelles within the cell. Still, others are carried to the membrane and secreted outside of the cell. So, I'm going to look again to kind of get it given away by the title of the paragraph, the Golgi apparatus. But that is what repeated a number of times in this paragraph, and kind of all the other information relates to that. So I'm going to say that this paragraph is about the Golgi apparatus, which moves proteins from the endoplasmic reticulum. And also, the Golgi apparatus sorts and delivers proteins, and within its membranes, it can change the shape of proteins. The Golgi apparatus is responsible for packaging proteins that can be stored, transported, or secreted. Okay. The next paragraph is also given a heading called vesicles. Cells need to separate reactants for various chemical reactions until it's time for them to be used. Vesicles, shown in figure 3.10, are a general name used to describe small membrane-bound sacs that divide some materials from the rest of the cytoplasm and transport these materials to the place within the cell. Vesicles are generally short-lived and are formed and recycled as needed. So I'm going to ask myself what this paragraph is about. So this paragraph is about vesicles. Vesicles are needed for different chemical reactions, and they separate reactants. They're small sacs made out of membranes, and they transport materials throughout the cell. Okay, last paragraph. After a protein has been made, part of the endoplasmic reticulum pinches off to form a vesicle surrounding the protein. Protected by the vesicle, the protein can be safely transported to the Golgi apparatus. There, any necessary modifications are made and the protein is packaged inside a new vesicle for storage, transport, or secretion. This paragraph is about what happens after a protein is made. Part of the endoplasmic reticulum cuts itself off and turns it into a sac to create a vesicle, and then this vesicle travels to the Golgi apparatus and keeps it safe. Okay, so we have gone through all 3 steps here by putting it into words on here. We've done the whole strategy: wrap, read, ask, and put into your own words. So you guys are going to do some practice with this on a passage from your textbook.